Friends, today we are going to learn about the new lecture, that is important characteristics of the subdivision Deuteromycotina, which is also known as fungi imperfecti. So what is this Deuteromycotina and what are the special characters of these fungi involved in Deuteromycotina? Deuteromycotina fungi are a group of fungi which reproduce only by means of asexual spores or fragmentation of hyphae or by modified mycelium. So the spores which are produced asexually are generally known as conidia. And what is this conidium? Conidium is a non-motile asexual spore which is formed at the tip or side of this sporogenous cells. Generally what we used to call these conidia are produced on the conidiophores. For this several genera of this group, that is under this Deuteromycotina fungi, they do not have any sexual reproduction or sexual stage, what we used to call as a perfect stage or telomorphic stage, which is not known for this group of fungi or not yet discovered in this group of fungi, or they have been dropped, that is sexual stages have been dropped from these life cycles during the evolution of these organisms. And these fungi are commonly called as imperfect fungi or mitosporic fungi because only the conidia is produced in this group of fungi, as there is no sexual reproduction, and technically they are called as fungi imperfecti because only have imperfect stage or conidial stages. And whenever the perfect stage used to be found, then based on that sexual fruiting bodies, they used to be placed generally under Ascomycotina or Basidiomycotina subdivision. Most of the cases the perfect stages which found to be belong to the this Basidiomycotina group. And coming to the vegetative structure, that is mycelium. Mycelium is well developed in Deuteromycotina fungi and used to be septate with branched hyphae and multinucleate cell. Multinucleate cell means each cell is having a more number of nuclei, hence we used to call it as a multinucleate. And this mitosporic fungi cannot be given hierarchical status above the rank of genus. Why we cannot give hierarchical status means because of the absence of sexual reproduction in this group of fungi. So since the present classification is based on characters of sexual stage, whatever the these fungi, that is Ascomycotina fungi, Basidiomycotina, Zygomycotina, how they divided these fungi means this classification is entirely based on sexual reproduction. But however the sexual reproduction is absent in the Deuteromycotina fungi, so this classification is not natural classification, this is artificial classification. Generally the classification of Deuteromycotina fungi we will refer it as a artificial classification. For most of these genera, perfect states are not known or very rare in the nature. They are temporarily grouped as members of what we used to call form class, form order, form family, form genus and form species, not generally as a class order what we referred in other group of fungi. Coming to the classification of Deuteromycotina, in this lecture I will explain about the various classifications available for the Deuteromycotina fungi, and description of the various genera I will cover in under coming lectures. So because if we will cover all the genera descriptions, it will go very lengthy lecture. So in this lecture we will see about the various classifications available under the Deuteromycotina. So this classification is completely artificial and based on only conidial peculiarities, as there is no sexual reproduction. Entire classification is based on type of conidia and how they are produced, and it cannot therefore be called as a natural classification. Already I told that it is not a natural classification, it is an artificial classification. And what are the main characters the researchers considered for classifying this Deuteromycotina are, that is presence or absence of asexual spores, that is conidia, and type of asexual fruiting body and the manner of production of asexual spores. So there are different asexual fruiting bodies like pycnidium, esservioli, I think already I covered about these fruiting bodies in my previous lectures. If you could not find about these fruiting bodies, just write in the comment box so that I will make a separate video about these fruiting bodies. And another one is the morphology, that is how this conidia is looking like, what is the shape of this conidia and the size of the conidia, and what is the color, and how the septation within the conidia of this asexual spores, that is conidial spores. So, presence or absence of asexual spores and type of asexual fruiting body and manner of production of asexual spores, that is how they are producing, in which manner these conidia are producing, and what is the morphology of these asexual spores. Coming to the first classification, that is classification was given by P.A. Sakado, and this classification we follow nearly 100 years up to from 1800s to 1900s. We followed this classification. What the P.A. Sakado did was, he classified the Deuteromycotina subdivision into 3 orders initially, that is the moniliales, which is also known as hyphomycetales, and second one is the melanchoniales, and third one is the spheropsidales. After proposing these 3 orders, then later Mycelia sterilia was included. How they classified the subdivision Deuteromycotina into 3 orders. Moniliales, he kept the fungi which form the conidiophores and conidia directly from the hyphae. These fungi he placed under the order moniliales or hyphomycetes. And coming to the melanchoniales, the name itself indicating that the conidia are produced in a saucer or disc shaped structure, generally used to call it as a acervuli. Under spheropsidales, he included the fungi, that is members which produce the conidia in a flask shaped receptacles known as pycnidia. Here under spheropsidales, what is the fruiting body? It is the pycnidia, and in pycnidia conidia used to be produced. In case of melanchoniales, fruiting body is the acervuli. In case of Mycelia sterilia, the name itself indicating that members which are sterile, that they do not produce any conidia. So, initially there were only 3 orders under the P.A. Sakado's classification, that is moniliales, melanchoniales and spheropsidales. And later this Mycelia sterilia, that is the 4th order, it was included. And these 4 orders again were divided into different families and sections. Suppose if you will see moniliales, it is divided into 4 families, and melanchoniales into 1 family, and spheropsidales into 4 families. Then Sakado divided each family into 7 sections based on septation and shape of mature conidia. Here in these pictures you can see the various shapes of conidia and their septation inside the conidia. So, what are the different type of those sections? First section is the emerospore, that is which are having 1-celled spores. Here you can see emerospores. And second one is the didymospore, which are having 2 cells, here you can see. And third one is the phragmospore, the name itself indicating that phragmo means fragmentation used to be there. 2 are many celled septa, here you can see the phragmospore. And fourth one is the dictyospore. Here you can see dictyospore, which used to be muriform spores, that is it possesses both transverse and longitudinal septa. And fifth one is the starospore, the name indicating that starospore means the spore look like star shaped. Here you can see this starospore. And sixth one is the scolicospore, that is conidia look like a filiform spores like thread like structures, here you can see. And last one is the helicospore, that is helical spores, here you can see helical spores. So, he divided each family under these orders into different sections based on the septation and shape of this mature conidia. This is the P.A. Sakado's classification. Then coming to the another different classification, what generally is to call Cusassian classification, which was proposed during the year 1953. This classification is entirely based on the origin of conidia. So the classification based on conidial origin is generally known as Cusassian classification. So what are the different type of spores are formed? So why this classification came into existence? So under this classification, different type of spore formation was mentioned. What are those different spores? First one is the blastospores. The name itself blastospores, that indicating that these are formed by simple budding, as we can see in yeast. And here you can see from this conidiophore, that is a mature zone of conidiogenous cells, they will blow out. Here at the terminal portion of this hyphae, some part of this conidiogenous cells, they will blow out and they will enlarge and develops into a conidium. Here you can see. And after formation of this conidium, a constriction will develop from the conidiophore or mature hyphae between the conidium, so that this conidia will separate from the conidiogenous cells before separation. So before separation, constriction formed between the conidiogenous cells and conidia. So after that, conidia used to be separate from the conidiogenous cells. What is conidiogenous cells? These are nothing but these are specialized cells which are formed from the formed within the hyphae, from which this conidia will develop. Where we can see these blastospores means in the species, in the genus Torula we can see Cladosporium, we can see, and Bispora and Septonema. In these four genera we can see this type of blastospores. And coming to the second one, porospores, the name itself indicating that some pore like structures are formed. So a pore is enzymatically dissolved in the outer layer of the wall at the tip of conidiophore. Here you can see, here outer wall is enzymatically dissolved to form the pore like structure, and inner wall it came out to form the porospore, and after that some constriction used to be formed to separate this porospore from the remaining this conidiogenous cells or conidiophores. We can see this type of porospores in the Alternaria, Helminthosporia and Curvularia. Coming to the third type of spores, that is phylospores, the name itself indicating that these spores are formed on specialized structures known as phyalides. So what is phyalide? Phyalide is a unicellular structure. Here you can see these phyalides, two different type of phyalides. Here we can see this is a unicellular structure, that is it is a single cell which used to be either oval, subcylindrical or flask shaped with distinct basal swelling and narrow distal neck. Here you can see here basal swelling and here narrow distal neck, and it is borne terminally on simple or branched conidiophores. Here either phyalides may produce simple or branched conidiophores, they used to be produced, and from the tip of these phyalides, these conidia arise either exogenously or endogenously. And here we can see two types of spores, one is dry spores and one is slimy spores. In case of dry spores, the conidia used to be in the formation of chain. That is conidia used to be formed in the chain, and we can see in Aspergillus and Penicillium. And second one is the slimy solitary spores. Here in second picture you can see slimy solitary spores, where these spores used to be adhere together in a slimy ball like structure. These slimy solitary spores we can found in Trichoderma and Stachybotrys. So fourth one is the gangliospore. Then the gangliospore formation is also looking like similar to blastospores. In gangliospores what will happen? The conidium developed by transformation of swollen tip of the hyphae into a pore. Here this is the hyphae, where tip of the hyphae used to be swell and it form a conidia like structure, which we used to call as a gangliospore. Only difference between this blastospore and gangliospore is that in gangliospore entire this conidiophore apex used to be transformed into a conidia like structure, whereas in blastospore spore only a narrow portion of the conidiogenous cells or tip of the hyphae transform into a conidia like structure, and in blastospore constrictions used to be formed, that is to separate the conidia, but here in gangliospore there is no formation of constrictions. Here we can see in the picture there is no constrictions here. And the last one is the arthrospores. Arthrospores is the formation of spores directly from the hyphae through the fragmentation. The spores which are formed through fragmentation from the hyphae generally used to call as a arthrospores. The term arthrospore has been used for spores which are resulting from breaking up of the hyphae into the cells. So with this hyphae used to be break into a different cells. Those cells generally used to call it as a arthrospores. There is no conidiogenous cell, but only a conidiogenous hyphae used to be have, and that conidiogenous hyphae also used to be some determinate growth. It will not grow indefinitely. It used to be grow definitely, and after certain growth this arthrospore formation used to start. Conidial initiation begins only after the growth of the hyphae has ceased. Already I told, after finishing this growth of the hyphae, then conidia formation used to be there. Where arthrospores formation we can see in the genus Harmodendron and Geotrichum. In these two genus we can see the formation of these arthrospores. So these are the different type of spore formation in Deuteromycotina fungi. Coming to the third classification, that is Ainsworth classification. Generally most of the people still nowadays we are following this classification only, who proposed during the year 1973. The Ainsworth classification is he divided the subdivision Deuteromycotina into three form classes, that is one is Blastomycetes, and second Hyphomycetes, and third Coelomycetes fungi. Under Blastomycetes, he further divided into two form families. One is Sporobolomycetaceae and second one is the Cryptococcaceae. Under Sporobolomycetaceae, generally ballistospores used to be produced, whereas Cryptococcaceae we cannot see any ballistospore formation. And Sporobolomycetaceae, the fungi generally used to call as a mirror picture yeast or shadow yeast. They used to form, if we will keep the leaf sample on the growing media, generally exactly the fungi will produce the mirror image of that sample. That's why generally we used to call it as a mirror picture yeast or shadow yeast. And Cryptococcaceae, we are having genera like Candida and Cryptococcus. Candida albicans we can know very well. This is the first fungus reported to cause disease in man. Whereas in Cryptococcus, generally this fungi used to grow on bird excreta and workers cleaning places. We can see this Cryptococcus fungus. And he divided the class, that is form class Hyphomycetes, into four form orders. One is Moniliales, which is also known as Hyphomycetales or hyphales, and second one is the Tuberculariales, and third one is the Stilbillales, and fourth one is the Agenomycetales. In case of Moniliales, distinguishing feature is that conidiophores, they will not fuse to form some specialized structures. Conidiophores used to be very free, and this under Moniliales there are two families are there, Moniliaceae and Dematiaceae. This is based on color of the spores, that is color of these conidial spores. They divided into two families, Moniliaceae and Dematiaceae, and conidiophores are also free. If conidiophores united into a fascicle like structures, they are coming under the form order Tuberculariales and Stilbillales. Under Tuberculariales, family Tuberculariaceae, under Stilbillales order, which is having the family Stilbillaceae, that is cinemata structures we can see in Stilbillaceae and Agenomycetales. These are the four form orders. Coming to the Coelomycetes classification, that is under Deuteromycotina, the form class Coelomycetes is further divided into form order Sphaeropsidales and Melanchoniales, and Sphaeropsidales is further divided into four form families. One is Sphaeropsidaceae, Excipulaceae, Nectroidaceae, and Leptostomataceae. So I will explain description of these form families, each form family and each genera in the coming following classes, because here I am not going to explain Just I will give the outline of this classification of this Deuteromycotina. In melanchoniales, we are having one family, Melanchoniaceae. Under melanchoniales, important fungi like Coleotrichum, Gliosporium, Pestaloceopsis, Pestalocia, these fungi will be these fungi are placed under the melanchoniales. And coming to the fourth classification, which is universally accepted classification, this classification was proposed by Kendrick and Carmichael during the year 1973 and Hawksworth et al. During the 1995. They divided the subdivision Deuteromycotina into three categories. One is imperfect yeast, which include the genera Candida, Cryptococcus and Sporobolomyces, and another one Hypomycetes mytosporic fungi. It includes the genera Alternaria, Botrytis, Cercospora, Curvularia, Fusarium, Helminthosporium, Lemoniara, Monilia, Nigrospora, Pyroclaria, Rhizoctonia, Trichothecium, Verticillium. Whereas acervular and pycnidial fungi, we are having two categories. Acervular, which were formerly included under the melanchoniales, that is Coleotrichum, Pestaloceopsis and Spaciloma, and pycnidial fungi are the Ascochyta, Botriodiplodia, Diplodia, Philosticta, Phoma, Phomopsis, Septoria. All these fungi are placed under the pycnidial fungi, that is acervular and pycnidial fungi. Under this classification, we used to study each genera as a description, and separately we will study. We will not combine into different families. But anyhow, in the coming classes I will explain about the Ainsworth classification only. In that classification also we will cover about all these genera and their pictures and their diseases caused by each genera. So thank you for your keen interest in this video, and remember to like and subscribe for more videos. If you subscribe to my channel, it will boost my interest to make it more videos, and you please share with your friends, because this is very informative for preparing competitive exams also. I wish you great success, health, prosperity and happiness. Thank you one and all.